Good morning, everyone. Welcome to the NanoOpen Webinar Series. First, I'd like to give a brief information about the project. NanoBio4Can Project, a mobility scholarship program for postdoctoral researchers, aims to develop nanobiotechnologies for innovative therapeutic approaches in cancer. While Sunum is the coordinator of the project, Izmir Biomedicine and Genome Center, Koç University Research Center for Translational Medicine, and TÜBİTAK MAM are the implementing organizations. For detailed information, please visit nanobio4can.net. Today is our third webinar. We have Dr. Müge Özkan and Dr. Noor Hamizah Binti Suhaimi from IBG, Dr. Nemany Abdelhamid Nemany Hanafy from KUTTAM. Each researcher will speak for 15 minutes, followed by a 5-minute Q&A session. We will organize this monthly webinar within the framework of the MSCA COFUND Grant Agreement, dissemination and communication obligations. This webinar will be recorded. It may be posted on YouTube. If you don't consent, you may keep your camera off. Dear all, welcome once again to the webinar. Thank you again for your valuable contributions. We will start with Dr. Müge Özkan. The stage is yours. Would you introduce yourself to us while starting the webinar?
Good morning, everyone. I'm Müge from NanoBio4Can Fellowship Program, and I'm a postdoctoral researcher in IBG. So I will share my screen. Should give me a second. I think you can see it clearly. Yeah. Okay. So let's start. So I'm Müge, and it will be a delight to meet you today. I will be talking about my project, CpG-TAT, nanoparticle treatment-based, nanoparticle-based mucosal treatment strategies in in vivo lung canceroma model. So before jumping into the project details, I just want to give a little bit information about myself. I'm a molecular biologist, traditionally trained later in immunology. I completed my PhD here in IBG under supervision of Gerard, and we investigated the role of immune NK cells in eosinophilic allergic asthma versus neutrophilic hard-to-treat Th1-driven asthma. Then after my PhD, I moved to Germany. Sorry, I should do. Yes, this is better. Sorry. I moved to Germany, and there I studied how early life infections, specifically pulmonary viral infections, shape the lung microenvironments. Then later, I moved to US to work with Lee Quinton in UMass Medical School, which I really appreciate the opportunity that Lee giving me. And I learned a lot. We studied lung-liver axis in hospital-acquired pneumonia. So as you can see, I've been working on pulmonary diseases and lung immunology right now for more than 8 years. And after US, I come back to home. And no surprise there for my project in NanoBio4Can is related to another lung diseases, lung cancer and immunology. So to give a little bit background information, solid tumors are challenging to treat, and the numbers agree. 65% of the patients who have a solid tumor in their colons, pancreas, and lung acquire resistance to immunotherapy. And when you focus to the non-small cell lung cancer patients, this number goes up to even more, to 70 to 85%. And one of the underlying mechanisms of this resistance to immunotherapy is immunosuppressive environments, tumor environment. So once tumor establishes itself to the tissue, it creates a protective barrier per se that limits and restricts immune cell infiltration. And even the cells that can manage to penetrate this kind of barrier, they have a reduced cytotoxicity. And as a cascade, it follows the poor treatment response. Then, due to that, the tumor elimination kind of reduced or fails, and you see a tumor persistence, thus results in a limited treatment response. To overcome this challenge, there is a newer strategy called in situ vaccination. So we just passed the COVID era, and probably we all know about the vaccines. So when you think about the traditional vaccines against infectious diseases, you have an antigen component of the vaccine that is derived from the microbe, and an adjuvant portion, which is the part that stimulates the immune system. So ISVs mainly have kind of this immune stimulation part, and they can be checkpoint inhibitors, co-stimulatory molecule agonists, nucleic acid, pathogen recognition receptors, immune cell itself, and or soluble proteins and cytokines that have an ability to give the immune system a push. And the important part is when you administer this ISVs, you give them to the local kind of area that you're interested in. So in our case, to the lung tumor environment, so that this kind of bump up or of activation in the immune system locally will kind of give this transient immune system activation that will cause some kind of killing and elimination of tumor cells. This will produce and increase the tumor antigen availability locally that later induces the generation of tumor-specific T-cell responses in populations, and hopefully the memory population. So when you ask why we can use ISVs, why it is kind of advantageous, the one thing is there is no need for tumor-specific antigen identification or engineering. Also, another trick is that when you give them these ISVs via mucosal route, it will create local response and tumor-specific, more like bigger and efficient immune response, hopefully, due to the resident immune cells in the local mucosal tissue. Another kind of plus is that for lung per se, when we say giving mucosal ISVs, we will use nasopharyngeal or oropharyngeal way. So it will eliminate this kind of needle treatment and easy to administer, like when you use an inhaler for the asthma patient, you just inhale. And also for a broader perspective clinically, it will have an opportunity to eliminate these big side effects that you see with the traditional treatments like chemotherapy. Excuse me. For our project, we will be using this kind of nano structure called nanoring that is composed of KODN, nucleic acid, and TAT peptide, which is a cell-penetrating peptide. When they combine together, they kind of form this nanoscale nanorings. And Billy Ginger, my currently scientific partner, we can call, and our at the time supervisor Marida Gürsel published that these kind of CpG ODN nanorings are actually immunostimulatory. They induce a type 1 immunity and can be effective in cancer treatment. So in our case, in our project, the idea is this immunosuppressive environment created by the tumor itself kind of blocks and reduces the efficacy of the anti-tumorigenic response and treatment. So we wanted to make it shift the balance to immune active by this in situ vaccination mediated tumor microenvironment tuning. So how we will do that? This is kind of the 3 blocks and pillars of the project. Is first the modeling of orthotopic lung tumor, which means that we will actually model the lung tumor to grow where it belongs in the lung, then treat it with nanoring and evaluate its efficacy by physiologically progressing tumor, monitoring the tumor growth, welfare of the mice, as well as the immune dynamics by checking resident cell, focusing on the cytotoxic response, NK cells, CD8 T cells, as well as their spatial distribution within the lung. So to check the model first, we use Lewis lung cancer cells that are luciferase stacked, which enable us to monitor the tumor progress with in vivo imager. We inoculated them mucosally, and we wanted to see whether we can be able to establish orthotopic tumors, monitor the progress of the disease reliably, calculate the tumor burden, and found the endpoint to determine a specific and efficient timeline to administer the treatment. So I'm proud to say that we actually successfully established our orthotopic tumor model, and this was a really very new kind of approach. Aspiration-induced orthotopic lung cancer. As you can see, we found out that if you give them the mice, inoculate the cells with aspiration, we can see 100% inoculation of the tumor, and we can reliably kind of follow it with the bioluminescent imaging. And the endpoint, human endpoint for the tumor-bearing mice is up to 30 to 40 day post-inoculation. So the second part is the treatment part. Right now we are doing it, giving nanoring in mucosally. So we tried 2 different routes, again, intranasal and aspiration, and we found that intranasal administration of nanoring is much more efficient. And we are right now collecting day 3, day 14 post-treatment samples to analyze immune dynamics and evaluating, as I said, the tumor growth and the immune dynamics. Right now, we are at the 17th month of the project, and I am close to wrapping up this efficacy of the nanoring treatment and the kind of repopulation of the level or concentration of the cytotoxic immune response. And I would like to thank NanoBio4Can team. This is kind of like we had excursion, and we meet at Istanbul for Industry Day at Trust Life. This is one of the rare occasions that we actually come together, which made me really very happy to physically see all the fellows and Meral Hoca and Didem. Then I attended immunology congresses, and last April I presented my method side of the project in a National Immunology Congress in Bursa. We also submitted our method article, and we foresee that for the whole project, I will wrap up the experiments around end of August and able to maybe send the paper in October, hopefully. Fingers crossed. This is our team. My science research partners. Bilgi, İrem is our technician, Buse our master student, and Dicle is our fellow master student. I would like to also thank all the people, technicians, specialists who are working in the core facilities, and thank you all for listening. And if you have any questions, I will be happy to answer. Thank you.
Thank you, Dr. Müge Özkan. Now session is open for question. You can just speak up or raise your hand, or if you like, write your question on the chat box.
Okay, I have a question.
Yes, please, Miza.
Okay. As for that, I mean, like you are already done the fellow up until 1.5 years. Do you find any challenges for the whole? Do you find any challenges for your whole study in these terms? That's it.
You mean for the modeling of the tumor?
Yes, yes. I mean, in terms of facilities or timeline of everything.
So thus far, our model works okay. And to be said, the only technical problem we had is with the tumor cell growth, because they are a kind of mixed population, and you have to be sure that they reach to a certain level so that you can inoculate properly. Excuse me. With the allergies, sorry. Apart from that, the inoculation, the tumor growths were much more homogeneous than when you compare with the other methods. And so we were talking about this non-invasiveness, because most of the orthotopic lung cancer models, they just directly inject the lung or do intratracheal administration, which are really very kind of invasive and requires a certain level of technical ability. So we wanted to make it more like homogeneous, and considering with the vivarium conditions and all, we wanted to make a minimum kind of invasiveness and more leaning on the kind of easy to use method, let's say. Does it answer your question? Okay, cool. Sure. Yep.
Yeah, hello. Thank you so much for your presentation. My question is about your nanoring. What about the size of your nanoring and its stability in tumor microenvironment?
Okay, that's a good question. So as far as I remember, 40 nanometer diameter ring structures.
40.
Yes, yeah.
And
4 nanoring.
And what is stability?
For the stability, once you prepare them, they will stay like, you can see them. So they have in the paper atomic force microscopic and SEM pictures, but I can say, because I didn't do this kind of stability tests, I just kind of, since we established, I used it. But there is a color change in the solution once they make the complexation, and it stays the kind of the physical appearance stays same for a week. That's longer, I put it. Like, there is no precipitation or anything. But we try to make it like prepare every time fresh, so that it involves like minimal risk. But for the tumor microenvironment, we didn't make any kind of, because we have to tag the nanorings. to visualize them in the tumor microenvironment. We didn't do that yet. We just want to first assess the efficacy and then move on to where they are with the immune cells.
Okay. Thank you.
Yeah, you're welcome.
We have a question on chat box, Müge.
Okay. Right now, maybe I can stop share because I can't see the chat
I
box.
can read it.
Okay.
Ali Sami asks, how do you track the size of tumor?
We track it with in vivo imaging. So it gives you a kind of ballpark size. I mainly use it at the first time to see whether tumor is inoculated. So it doesn't give you a really very sensitive results at the beginning when it's like tumor size is small. But after certain points, I found that day 26 after tumor inoculation, you see like the intensity of the photon kind of exchange gives you a really nice kind of expansion curve. But it's kind of relative. So we do this like every week. We get measurements for the bioluminescent imaging to track the growth in a per se. You cannot make like size. I can only do the tumor burden after I do like endpoint pathology, when I like sacrifice the mice, take out the lungs, and do the kind of sectioning with the digital pathology determined tumor versus whole line ratio.
Thank you, Dr. Müge Özkan.
You're welcome.
For your presentation. Dear Dr. Nemany, if you are ready, stage is yours.
Thank you, my professor. My name is Nemany Abdelhamid Nemany Hanafy, NanoBio4Can, MSCA COFUND Fellow at KUTTAM. First, I will show you a table of the content. I will talk about academic and the professional background, summary of our project, our challenge and our perspective, scientific content and our result, and then scientific outputs. So first, I am academic staff, associate professor, index 26. Nemany Hanafy has been awarded position as a scientific collaborator in Italian National Research Councils, and there he succeeded to get a doctoral degree and collaborating with Salento University. And then Hanafy achieved 2 scholar visitors, one in Paris hospitals, and the second in IPLL Barcelona. And Hanafy working in cancer care, and finally he worked in Kafr Sheikh University. Hanafy developed several approaches like hybrid protein nanoparticle, lipid nanoparticle, theranostic system, block copolymer, graft copolymer, and rare barrier technique. And now he is senior editorial team in scientific report. He is editorial board member in Discovery Oncology. He is edited research topic in Volunteer. He is selected as a topical advisory panel member of Polymer in MDBI. And Hanafy, he is the top 2% listed scientific over the world for last 3 years, to 2023, 2024, and 2025. So what about the summary of our project? We need to look for glycolysis and our cell relations. Glycocalyx is a highly charged layer, attached on the biological macromolecules and the bound with cell membrane. This glycocalyx composed of glucose aminoglycan, glycoprotein, and the protoglycan, appearing sialic acid in the terminal. So sialic acid is 9 carbon atoms structured in the core, and it has 3 configuration in acetylnormanic acid, in glycolyl normanic acid, and D-amino normanic acid. These sialic acids formed in cytoplasm and deactivated in nucleus by adding cytidine 5 monophosphate. This substrate moved to Golgi apparatus. Inside the Golgi, it is assembled to galactose by different enzymes. This enzyme is called sialytransferase enzymes. It is around 20 different enzymes, and the configured sialic acid on galactose by 4 configurations according to sialylated galactose linkage in alpha-2,3, or sialylated in acetylnormanic acid in alpha-2,6, or sialyl antigen in alpha-2,9, and finally sialylation linkage in alpha-2,8. So what is the relation between sialylations and our multiple myeloma? It is found that during hypersialylations, the molecule stromal-derived vectors 1 increased, and it is receptor increased as well. So these 2 molecules increased adhesions of multiple myeloma in bone marrow via fibronectin and the vascular adhesion molecule. At the same time, once hypersialylation increased, P-selection glycoprotein ligands increased, and the drug will be resisted. Also, during hypersialylations, immune system cannot attack multiple myeloma completely because sialic acid block cyclic 7 and cyclic 9 on the surface of natural killer cells. So what we need to inhibit sialic acid on the surface of multiple myeloma to allow natural killer cell to attack multiple myeloma. So to do that, we have some challenge. First, in previous literature, there is inhibitor is promising and inhibited exactly the sialic acid expressed by this. First, this inhibitor is pre-acetylated 3-fluoroaxone neuromanic acid. It is acetylated hydroxyl group by OAS, and the added methyl ester instead of OH, and the added fluoride, axial fluorine, and carbon number for this molecule. So sialic acid is inhibited. But sorry, if it is a promising molecule but cannot pass clinical trial, because after injection for 54 days, it is cause kidney failure. So this molecule not working completely on a clinical trial. So we need to look for other biological and biodegradable molecule. So our project work on in this area. The second challenge I saw in this project is when I looked for biodegradable molecule is soya saponin, but so much expensive, 5 milligram, around $500. Also, if we need to isolate, we need intensive isolation technique to just isolate few amounts of concentration of soya saponin, and we need purification, higher purification. Second, I worked on multiple myeloma that already is suspended cell lines. It cannot go for 3D spheroid, so we need to find solution to solve these problems. First, I isolated soya saponins, and we looked for its structure compared to our standard, and we find exactly the molecular weight by mass spectroscopy, and we identify the chemical structure by NMR. So it is similar to traditional soya saponin in the market. I get this soya saponins, and I try to do chemical competitions by simulation to understand what is going on. It it attached completely when I get our cell transferase, and I moved the pocket, and I reacted the pocket with our cell transferase, with our inhibitors. It is attached on the, completely with the pocket and inhibited the pocket according to simulation of the chemistry. So the first challenge was solved. Second challenge, multiple myeloma is suspended cells. So we need to do 3D spheroid in these suspensions. I did 3D patches, and I injected cell lines inside the patches. This patches matrix, and I allowed to grow. So it is growing in 3D spheroid structures. And now we have a model for multiple myeloma, can be used to study metastasis, can be used to study migration and the deviations. So this model is new, and we applied for patents on these models. So our aim now to target nanoparticle delivery of sialyl transferase inhibitor for treatment of multiple myeloma. My project completely working on bone targeting liposomes, but when I came here, I divided the project for 3 chapters. First chapter, multi-drugs and diluting capacity, seronistic cancer bioimage, and liposomal bio-nanotechnology. A multi-drug and diluting capacity, we encapsulated photodynamic therapy, chemotherapy, anti-sialyl transferase inhibitor. Seronistic cancer bioimage, we did characterization and defabrication. Liposomal nanotechnology, I started to finish this area in the current days. So our multi-drug and diluting capacity, we did capsules with 25 nanometer. We encapsulated sialyl transferase inhibitor in A, and we encapsulated sialyl transferase inhibitor and chemotherapy in B. We encapsulated sialyl transferase inhibitor and photodynamic therapy in C, and we collected all of them in D, photodynamic therapy, chemotherapy, and the sialyl transferase inhibitor. We saw cellular uptake to understand nanoparticle accumulated inside the cytoplasm and an area of pre-nuclear regions. So we're looking for inhibition of sialic acid. We find our material inhibited sialic acid. If it is sialic nanoparticle with sialyl transferase inhibitor, or sialyl transferase inhibitor with CBT, and the sialyl transferase inhibitor with RP, and the sialyl transferase inhibitor with CBT and the RP. But when we irradiated, the sialic acid started to be overexpressed again. We need to understand what is going on. I did fluorescence biotechnological probe. This probe is specific attached with sialic acids, and we get the image with confocal microscopy. Our inhibitor is inhibited sialic acid alone, and also with CBT little bit overexpressed, but in RP or photodynamic server is inhibited, and all of molecule is inhibited also. One is irradiated, we find sialic acid started to be overexpressed, and this result is positive with previous published papers that say during irradiated sialic acid increased, and this go for metastasis. So we did seronistic cancer bioimage by carbon quantum dot and our material. We confirmed the result by atomic force microscopy, transmission electron microscope, mapping, and EDX. And we did engineering for carbon quantum dot by adding metals, and this overexpressed the metals on EDX curve. We find cellular uptake by intensity of this carbon quantum dot on our cell lines, and also this material inhibited sialic acid compared with the other inhibited sialic acid in this area. So according to our scientific output, we published 2 abstracts in our conference last times, and we did the review. I did collection issue for innovative nanotechnology of multiple myeloma. We applied for 2 patents, one for purification and the characterization at the we are going to finish this ones, and they applied for 3D patches patents. We have conference, an international electronic conference on cancer. So finally, I would like to thank NanoBio4Can to provide me this great opportunity to work with scientific papers. I would like to thank Koç University to provide me this facility and the KUTTAM department. I would like to thank my professor, Safajan Kurman, and thank you so much.
Thank you, Dr. Nemany. Now Q&A session is open. You can just speak up or raise your hand, or you can use chat box.
Maybe I have a question. So actually, like regarding the theranostic bio, do you hear me well?
Yes, we can hear you.
Yeah, I'm asking about the theranostic bioimaging. Could you please like talk more about the theranostic bioimaging? How do you use like your combination in order to do like the bioimaging?
Please, could you repeat your questions again, because I didn't hear you well?
I'm talking actually about the theranostic bioimaging, and you said like actually you use your nanoparticle in order to do like a bioimaging for the theranostic applications, right? So could you please like talk about that again, because I didn't get that well.
Okay, thank you so much for your great questions. Theranostic, it means I have a molecule can be give me diagnosis, and in the same time I give treatment. For carbon quantum dot, because it is very smaller diameters, we can use it as theranostic cancer bioimage. But because carbon quantum dot should be engineering a little bit to improve the intensity of the intensity of the carbon quantum dot, so we have used some metals to improve the structure of carbon quantum dots and crystal building crystals of carbon quantum dot to give this indication. Carbon quantum dot sometimes the toxicity is higher, so we have to focus to target carbon quantum dot for cancer cells. So I did ligands on the surface of carbon quantum dot during fabrication, so it is targeted cancer cells. And cancer cells, we find this carbon quantum dot has ability to give us apoptotic inductions, and in the same time give us emission for intensity without using fluorophore. Is okay?
Yeah, thank you. Maybe like we could follow up again, like with that. I mean, on live or something. Thank you.
Oh, thank you.
Any questions? Thank you, Dr. Nemany.
Thank you so
Your
much.
presentation. Dr. Noor Hamizah, if you are ready, stage is yours.
Thank you, Nokta. So can you hear me very well right now?
Yes, we can hear you.
Okay, can you see my screen right now?
Yes.
Okay, so good morning, everyone. Thank you, moderators.
I start your time.
Okay, thank you, moderators. Good morning to all the professors and my fellow friends. Thank you for joining me to this webinar. Maybe I'm the last speaker. I hope you still have some energy to listen to my some talk. And my name is Noor Hamizah. I am one of the fellow programs that are held in the IBG. So I will just today I will just present my current progress research during my time at IBG. But before that, let's talk about a little bit about myself. My academic journey began when I got my PhD in Japan, in Shizuoka University, for around 3.5 years, and then I moved to Malaysia after my finishing PhD to do a postdoctoral in Malaysia, and then after that I got a better offer in A-Star in a proteins analytic group, so I moved there for about 1 year, and then after that I saw there is like a very good opportunity in this fellow program, so I moved to Turkey for this program. So all above this time that I spent is all about to establish my research field, like you see in the screens. So let's just directly to the main of the topic of my research, which is modulation of O-glycosylation in Pichia pastoris for anti-PD-L1 antibody production in cancer immunotherapy. So along the presentation, I will just begin with a brief introduction to the PD-1 and PD-L1 interaction and why it is important in the cancer treatments. And then I will explain the research objective and the research scopes, followed by the implementation plan. And after that, I will summarize the result and several achievements so far. And I will just conclude with the challenges and future work, and also the training and dissemination. So to begin with the scientific background, this slide show the mechanism of action of the immunotherapy of the PD-1 or PD-L1 axis blockade. So if you can see from these first figures, you can see that under the normal tumor immune evasion condition, PD-L1 on the cancer cells will bind to the PD-1 at the T cells, and it will suppress the activity of the T cell to kill the cancer cell. So this is why it is important when this interaction is blocked by the anti-PD-L1, where this T cell, after it is being blocked by the anti-PD-L1, this interaction will make the T cell still remain active and recognize the cancer cells and attack the cancer cell more effectively. So this is why anti-PD-L1 antibodies are important therapeutics molecule in the cancer immunotherapy, and this is also why we are trying to produce it in our laboratory. And with that biological background in your minds, I will now explain why I chose Pichia pastoris as a recombinant protein that I use to produce in these studies. One of the reason is because it is grow easily and fast to produce, and it is also have a free of endotoxin and viruses, and it's easy to control manipulation of the yeast expression, and then it have the diverse of the post-translational modification. It also will easier to purify the secreted protein from the growth medium. This is very important in industrial because we really want to avoid the host cell protein from the from the intracellular cell. That's why it is really important to have the protein that are secreted outside from the cell, and it also have the high level of productivity. However, due to the diverse of post-translation modification here, it is come to our problem statement where the protein that are produced in the Pichia pastoris have the addition of the glucose here. So this is very important because in industrial also they have a major challenges to face this problem because they couldn't produce a very clean and uniform antibody from the anti-PD-L1. So this is current research focused on the glycoengineering strategy to control and to standardize this modification to achieve the uniform and clean antibodies. So based on these challenges, so the project was designed into 3 main work packages, as you can see here. So this slide shows all about the research objective and the research scope. So the main goal of the project is to modulate, as I already explained earlier. So according to this research objective, so the work is divided into 3 research work packages, which include brain engineering, expression and optimization, and glycosylation. Plus also we want to see the functional quality of the antibodies that produce during this engineered stream. So we go directly to the implementation plan. So this implementation plan are across 2025 and 2026. So in the main of 2025, we are focusing on the work package 1, 2, and 3, which are focused on the gene cloning and plasmid construction, and followed by the optimization of the expression condition, and also the purification and characterization. However, work package 3 is continued until 2026, and we are trying to compile all the data and writing within these years, and we are trying to submit one paper at the end of this year. So these all the timelines show the logical progression from the strain construction, expression studies, and finally to purify and characterization for the produced antibodies. So this is the slide. I will move from the plan to the progress I achieved so far. So this slide is summarized and achievement from the work package 1. So the completed activities include the CRISPR-Cas9 knockouts of the engineered strain, and then followed by transformation and screening, and followed by the strain growth and characterization. So one important achievement so far from this work package is the poster presentation on the strain development of the Pichia pastoris with the knockout strains through the CRISPR-Cas9 modulation editing gene, and it was presented in the 23rd Biotechnology Congress in Ankara. So this slide, the next slide will be about the next work packages, where we are going to observe the antibody expression with and without the overexpression of the protein disulfide PDI. So this slide, currently we have like 3 sub of the work that need to be done through this work package. First is the quantification of gene copy number of the overexpression PDI. So from this result, we can see that we are trying to get the highest number of gene copy number that are integrated in that modified strain of the Pichia pastoris in the work package 1, so that we are going to use this high copy number to do some integration of the antibody expression of the MT-PDL1. So basically, we are trying to do step-by-step of the expression by using the 96 deep well plate and the SHIP plus studies, where from the 96 deep well plate, you can see we can find the best colony that can express the heavy and light change of the MT-PDL1. So we are trying to improve this expression in the SHIP plus. So from the SHIP plus, you can see there are several strains that have improvement in the expression of the heavy and light change. And also you can see that there are also has like some strains that have the how to say, like a multiple isoform appears here, and some of other strains have a very clean antibodies producing. So this is really I mean, it is really unexpectedly predicted. So from this result, we also are trying to do some purification of the antibodies through the protein A ACTA column, because from these studies, we can get the purified sample to further characterize the other sub-work package for the characterization and functionality test. However, from this data, we can see that we can get the good coverage of the binding with the protein A ACTA column. So it is really good enough for the next study results. So in the last work package 3 is we are trying to characterize the O-glycosylation profile and the functionality quality of MT-PDL1 in engineered Pichia pastoris strain. So if you can see that the first part, we need to find the O-glycan profile using the purified MT-PDL1 as in the work package 2. So from that data, we can directly use for this characterization. So in this O-glycan profile, we are trying to do in the 2 method, where the first method we are using the enzyme method, And another method, we are trying to deep analysis in the through the LCMSMS. Maybe we will do in outsourcing to the others expertise, so they will help us to fully characterize the O-glycan profile and predicted the structure of the O-glycan that are produced in our anti-PD-L1. So after we got all the results, we are trying to do the functionality of the purified anti-PD-L1 based on the ELISA. So this is important. If you can see, this method will do like we are trying to see whether our purified antibody PD-L1 have the very good functionality or not with the binding with the PD-L1. And from this result also, we are trying to see the apparent KD value, whether our anti-PD-L1 can produce the good apparent KD value or not. So it is showing whether our antibodies will retain its functional binding quality or not. So through that, all the work package 1, 2, and 3 that of course have the current challenges that I'm facing throughout this study, where the first one is about the complexity of the O-glycosylation, where it is really unpredicted result is appeared. So we need to have like sometimes to make some strategy again to face this problem. And also we are facing with the analytical limitation in IBG itself, and also in the Turkey itself. So it is really trying to find some expertise outside from this country or inside this country that can give give the how to say, like advice how to do for this analytical experiments. And of course about the glycosylation and functional relationship, because without the data from the analytical parts, we cannot proceed with the functionality relationship. So for the future direction, maybe we can do here, or maybe we can do some continuation in another way, where we need to have the further exploration on genetic strategies, and deeper detailed analysis on glycosylation profile is needed, and then functionality validation to confirm the therapeutic relevance, such as like uptake studies or some kinds of like animals vaccination studies to see our functionality of anti-PD-L1 is really working or not. And for the last part of the how to say this webinar, I would like to show our I would like to say that I go to several training and dissemination, and it really helps a lot during the program, because for example, like training from the ACTA chromatography in Civitas is really helpful for me for my experiments. So I'm really happy with that. And also I attend some congress in Hajjat University, and it also really good for the diversity in the research itself. And I contribute some of the articles in the NanoBio4Can platform. So all above these activities are important because they can support both technical developments and broader the research dissemination. This also can help to strengthen the collaboration, the visibility of my research, and also my scientific effect. And last but not least, we come towards the acknowledgement, of course, to the NanoBio4Can program itself. Without them, we cannot done this program successfully. And my principal investigator, Nemany, and the co-principal investigator, Dr. Siddharth, and all my FG laboratory staff and members for the present and past, especially this person, because without them, I cannot have the smooth sailing in my research in IBG. So that's all for me. If you have any questions, you can ask me. Thank you.
Thank you, Dr. Noor Hamizah Suhaimi. Now the Q&A session is open. You can just speak up or raise your hand, or you can use chat box. Noor Hamizah, I think you have a question from Ali Sami in chat box. How many routes you said you used to for
No,
the...
no, that one is from Müge, I think.
Okay, sorry.
I think we don't have any question, but maybe I will ask some of my friends for my to give some recommendation or some feedback to me privately is also possible. Thank you.
Noor Hamizah, there is a message on chat box.
Oh, for Senthil.
From
Okay,
Senthil.
Senthil. The challenges that I faced during the antibody purification, of course, there is first I'm using the manual purification using the column. However, it doesn't have the good coverage binding, so that's why I'm moving from manual column to the ACTA, protein A ACTA. So from the protein A ACTA column chromatography, it really has the good coverage, and that's really helpful for me for the purification.
Is there any question? Thank you. Thank you, Dr. Müge Özkan. Thank you, Dr. Nemany Abdelhamid Nemany Hanafy. Thank you, Dr. Noor Hamizah Binti Suhaimi. And thank you all participants for your participations. The fourth webinar in our series will be held on 3rd of June. Dr. Saif Ullah Afridi, Dr. Senthilkumar Palanisamy, Dr. Shoaib Anwar Ansari will present information about their research. Thank you again for your participation, and goodbye and good luck.
Thank you.